Okay, so here comes the word somatic cell again, for those of you who felt unfamiliar with it before. We are going to talk today and tomorrow about the cell cycle of somatic cells and something called mitosis. So today we're going to cover the content. Tomorrow we are going to do, like I said, a virtual lab. Now, it's in the title, but what we're talking about today is a cycle. And so that means it can happen over and over and over again. And I'm pointing this out because what we'll talk about in lesson 3 is not a cycle. And so it's a distinct thing that makes it different. Now, this is just one picture that represents the cell cycle. I picked it because I liked the colors that were on it, I think. Your textbook, if you look at the digital textbook, has a slightly different looking picture. And we will go to the library when we have our break in a few minutes for anyone who wants to check out a textbook. Now, a somatic cell, just to reiterate, is a body cell, not one that is making a gamete, but any other body cell. Soma means body. And so like I said before, it could be brain cell, bone cell, muscle cell, heart cell, liver cell, whatever. Any other cell in your body should be going through this cell cycle. Now, underneath this, I put something that should be sort of familiar if you think way back to Science 10. You probably remember learning about surface area to volume ratios in grade 10. You probably measured something. Depending on who your teacher was, I know I used to get my class to measure these little blocks, or sometimes I do it with Jell-O cubes. But measuring something and figuring out what its surface and what its volume is usually something you do in grade 10. And the conclusion that hopefully you drew in grade 10 was that there's a point where cells are too big. They have so much volume on the inside of them that there's not enough surface on the outside to maintain their life, so they have to divide. Now, that's a Science 10 idea, but it's why we have to talk about the cell cycle, okay? So the idea is, if an organism wants to get bigger, it needs more cells. Not bigger cells, more cells. So we need a way to make more cells. And that's what the cell cycle does for us. Now, if you look at the picture, the large, vast majority of the cell cycle is interphase. So see this whole gray section here that includes the light blue G1, the red S, and the, what color am I going to call that? Other blue G2. All of that is interphase. Interphase accounts for about 90% of a cell's lifetime. And then underneath, I'm telling you what happens, generally, in each of those 3 sections of interphase. The first section of interphase is G1. Now, in the olden days, the G stood for gap because scientists had no idea what the cell was doing here. Later on, we understood more about cells and we got better technology. So the G can stand for growth. I will not ask you what the G in G1 stands for. I just want you to know that's where it came from. In G1, cells are growing and cells are synthesizing proteins. Synthesizing means making, so that's what they're doing. Cells getting bigger, cells making proteins. Proteins are the building blocks of most things that the cell needs to do, so that's why it's important that it's making them. Now you see I've got some hours listed there. If we imagine that a cell's life is one 24-hour period of a cell's life, about 11 of them would be G1. These are just approximations. Not every cell will follow this, but it's just to give you an idea of how much of the cell's life is in that phase. The S phase stands for synthesis. That's what the S stands for. It's funny because the cell is doing synthesis in G1 and G2, but in the S phase specifically, the synthesis we're talking about is about DNA. In this phase, DNA replication happens. You'll remember in our notes on the first day, I showed you a chromosome, and then I showed you an identical copy attached to it. That's what happens in this particular section. That is when sister chromatids are attached at the centromere. So I went from having one chromosome to having two identical copies of that chromosome attached at the centromere. Now, on our 24-hour clock, this would be about 7 hours of that. G2, which is the last part of interphase, just like G1, the G used to stand for gap, now it stands for growth. Growth is again happening. We're still synthesizing proteins, but very specifically, we're refilling energy reserves. The cell needs a bunch of energy for this chunk that's going to happen next, because it takes a bunch of energy. And so the cell's getting prepared. So most of what interphase is the cell growing, making things, making copies of things, gathering up energy, so that it can do this little slice of pie here that we'll talk about next. Now, does anyone want to ask anything there about interphase? So, what does a cell look like in interphase? Well, here's a cartoony drawing of some things that you might see. You might remember me saying there is no point where you have to draw something realistically, especially on tests. I might ask you to draw something once in a while on an assignment, but on a test, I'm not going to require art as your answer. However, it is very probable that I would show you pictures and have you interpret them. So you're going to need to know what it looks like. So visually, if I were to draw a cell, notice a few things. First, there is an outer layer, and then there is that inner layer. Anybody know what the outer layer, the like darker blue, would represent? It's a part of a cell. All cells have one. Yes. Cell membrane. That is exactly what that represents. The inner sort of teal colored circle, you can see down here, oh, I'm not going to write there, that looks terrible, is the nuclear membrane. So the cell has a membrane around the whole thing, but there's also a membrane around the nucleus. You'll notice that I am omitting all of the other parts of the cell, really, except for the nucleus. I'm aware that cells have other structures and that they are vital, but for our purposes, when we talk about cell division, we're going to focus on the nucleus because that's the big deal for us. We want to focus on the DNA. Now, in interphase, the key features that we should notice are that chromosomes are not condensed. They look like strands of chromatin, and chromosomes are always found inside the nucleus. So most artistic representations of interphase will show squiggly lines, and you can't distinctively see how many chromosomes there are, how they're paired up. So you would look and be like, I don't know how many chromosomes that is. All I see are squiggly lines that are non-distinct. That's what chromatin looks like. Now I'm saying that you can see this here because we've drawn it in the picture, but realistically, if you were to look at this with a microscope, it would just look like a fuzzy gray blob. You wouldn't see anything distinct. The thing that is important, though, is that you would be able to see the nuclear membrane. Like, it would be obvious that there was a nucleus and that it was a distinct structure. And I'm pointing those two things out because in other parts of the cell cycle, those two things change. And it's how you can differentiate which part of the cell cycle the cell is in. Now, we're going to take a body break here. So, interphase happens in between cell divisions. That's why it's called inter. Inter means between. So between one cell division, which is that little piece of pie that's cut out, and the next, interphase happens. The other part of the cell cycle, even though it's the smallest piece of pie in the picture, is the one that we're going to focus on the most. There's going to be lots of detail here. So cell division is what we call that chunk there that's cut out. It's green, it has an M, it says mitosis and cytokinesis, and we'll talk about what both of those mean. But cell division is what we're focusing on. So we're going to talk about how the nucleus divides, that's nuclear division, and how the cytoplasm divides, that's the rest of the cell. Now the first three things that are listed there are the functions. This is why we need cell division. One of them I already alluded to, it's for growth. If an organism wants to get bigger, they can't just have humongous cells, that won't work. They need more cells. The second reason would be maintenance. Some of your cells don't last forever. Maybe someone remembers how long red blood cells last for? Sometimes people know that one. Yeah? You know?
I think it was like 120.
Yeah, 120 days. 120 days or 4 months, that is how long your red blood cells last. So they need to be replaced. That would be an example of maintenance. It's not like you injured yourself. Those cells just don't last forever and you need to make new ones. The third reason is repair. That's where some sort of damage has happened. So you're me and you cut your hand on something. I do this all the time, and I'll look at my hand and be like, why am I bleeding? I don't even remember cutting my hand on anything. Who knows? I need to repair. I need new skin cells to make up for the ones that I damaged. That would be repair. So these are the reasons that we need to have cell division. We need to make more cells so organisms can grow, so they can maintain, and so they can repair. Now, not all of your cells divide this way. For example, lots of cells in your nervous system don't do this. That's why brain damage is such a big deal, because the chances are if you damage a brain cell, it's not coming back. I don't want to say all nervous system cells because there is a lot of research, like a lot of research about regenerating nerves. And perhaps you're like, ooh, I like that. You might want to think about doing your de facto about that, about cells that we didn't think could regenerate, but maybe they can after all. The second point down here is that there needs to be a stimulus. Cells aren't just going to divide just because. If they do, we got a problem. If cells divide just because, you might have a tumor forming. That's how cancer starts, is when cells divide out of control. But there needs to be a stimulus. It could be a hormone. For example, we'll talk later in the course about GH, which stands for growth hormone. Chances are some of you are done growing, but some of you might still be growing, especially in height at this point in time. And that hormone could be telling your cells to divide so that you grow taller. The stimulus could also be damage. So something, you injured yourself and you need to replace those cells. Those are just a couple of examples. That is not an exhaustive list of reasons or of stimuli that could occur, just a couple of examples. Now, another topic that you might find interesting is aging. What does it mean to age? Sure, it means chronologically having more years go by, but biologically, we age when our cells cannot divide anymore. So we're going to talk about chromosomes quite a bit, and there are a couple of things that happen with your chromosomes that over time get smaller and smaller. There's things called telomeres. They are repeating segments at the ends of chromosomes. So at the end of each chromosome, there's like a chunk that repeats itself over and over again. And every time cells divide, those little repeated chunks get a little bit shorter, and a little bit shorter, and a little bit shorter. And eventually we get to a point where the repeating segment can't get any shorter. And what that usually means is the cell isn't going to be able to divide and replace itself anymore. Now you might wonder, but I thought men could make sperm for their whole entire adult life, which is basically true. They can. So how does that work? Well, there's an enzyme called telomerase. ASE is the ending typically for enzymes. So if you see a word that ends with ASE and you're in biology, it's probably an enzyme. I don't want to say 100% of the time because there might be words that aren't, but most ASE words are enzymes. It's an enzyme that we will find in sperm germinal cells, so the cells that are making sperm, that repairs the telomeres so that cell division can continue infinitely. We could figure out how to make this work on all the rest of your cells. Potion of youth, the fountain of youth, never-ending life, like the dude in the book that I was reading on the weekend who was like 3,000 years old. It was because of magic, though, not because of this. But that would be cool research to look into. Now, does anyone have something they want to ask here? About cancer, about cells dividing forever. Yes?
So, when you get cancer, is that basically like your red blood cells don't repair themselves?
So there's more than one type of cancer, but if we sort of say in general, what is it? Cancer is when cells bypass the stimulus. They just divide and divide and divide without anything telling them to. And it often means that they're doing it too fast and causing errors in the cell. And so you get all these cells, but they don't work properly. That's usually how it goes. Of course, there's differences in every type of cancer, but that is the general format for how a tumor forms. Anybody else want to ask anything there? If you wanted to do your de facto about cancer, please do not make the title be, did you know that there's a disease called cancer? Because yes, we do. You would need to pick something specific, like about a certain type of cancer or some cool new piece of research about it, but try not to be super general like that in your topics. Pick something specific. It'll help you focus your presentation. Now, we are going to talk about mitosis, the first chunk of cell division. So if I could I'll just go back one. See here, mitosis was first, then cytokinesis. They make up that little green section. Mitosis could be represented by the acronym PMAT, and acronyms are a good tool, especially in biology where there will be lots of steps that have to happen in a particular order. PMAT is the first letter of each of the 4 phases of mitosis. So for example, the P stands for prophase. Now, I picked the same artist's representations of cells as I used for Interphase. So you can see like the color scheme is kind of the same, but I really want to emphasize this is just one artist's drawing. And so not every piece of art looks exactly the same, which is why I'm always telling you with words what the key features are. So in prophase, which is the first part of mitosis, the first thing that you'll notice is that DNA condenses. That means instead of long stringy bits, it looks like a chromosome. It'll look like 2 copies attached by a centromere, these perfect little X's. If you can see the little X's, we're not in interphase anymore. The second thing that you can see is that the nuclear membrane decomposes. Now sometimes, like this picture, will show the nuclear membrane already gone, like you can't even see it, but it's decomposing as prophase is happening. So depending on whether it's right at the beginning of prophase or after it has completed, you might still see a bit of the nuclear membrane. So it might show up as a dotted line around the chromosomes. So there's more than one way that we could represent it, and I just want you to be on the lookout. Either the nuclear membrane is already gone, or it's in the process of being gone. The other thing that you should see are these things called centrioles. Here's one, there's the other. This artist drew them like little stars. Sometimes they look like 2 pieces of licorice that have crisscrossed over each other. They have varying ways of being represented. But what the centrioles are little structures that are going to help the cell divide its copies of chromosomes in half. Now, if you remember back and I'll just back up for a minute, to the interphase picture. See these two things? There were two little starbursts right there, and I didn't even mention them because they were not relevant during interphase. Well, now, those little starbursts, which are the centrioles, have migrated or moved towards the poles. Now, cells are round, if we're talking animal cells. So we tend to talk about them kind of like we talk about a globe, where there's two poles. We'll talk about things going across the cell, like we would with latitude and longitude. So we say poles, but we mean the two ends of the cells. So those are some things you should look for. Will every picture have all of these things in them? Maybe. Depends how the artist was feeling. But these are some things you should look for. Now, does anyone want to ask anything about prophase? So metaphase, that's the M in PMAT. Now the first thing you'll notice is that the centrioles, the little starburst looking things, are at the poles, and it looks like stuff is coming out of them. I always imagine Spider-Man shooting his webs out here. This is what makes me think of. The centrioles produce these little fibers, which are these dotted lines you see on either side. The fibers are made out of microtubules, so they're actually really tiny little tubes. And they form between the spindles. The spindles, if I can underline it, come from the centrioles. So the centrioles have these little structures that make these fibers. So if I was going to talk about these fibers, we usually call them spindle fibers because they come from the spindle in the centriole. Now if any of you remember Sleeping Beauty, she pricks her finger on the spindle of a spinning wheel and she's supposed to die and she only falls asleep. But it's the thing that generates the fibers. That's where the word comes from. Now, why do I need these fibers? Well, the chromosomes are going to have to move. I'm trying to make a new cell, which means I'm going to need to take some of the chromosomes and put them in one spot and some of them elsewhere. And so these fibers are going to facilitate that movement. And if you notice this point here, the fibers attach to the centromeres. So you can see that there's one fiber going to the centromere, that middle portion of each chromosome. Now, all of those things are great, but really the most distinctive thing I think about metaphase is how the chromosomes are lined up. Metaphase starts with M, so does the word middle. In metaphase, the chromosomes are lined up in the middle. So this cell is drawn with the middle horizontally, but cells are circles. So keep in mind that we could be talking about the vertical middle as well. This picture's bad because the smartboard doesn't like drawing where I'm drawing. But the chromosomes could be lined up horizontally or vertically. I could turn that circle any direction, and the middle would always be the middle. Now, here's something that people find hard even after we get through the whole chapter of lessons on this. Each one of these things is a chromosome. Each chromosome has an identical copy attached. The identical copy got made during the S phase of interphase. So when they're attached like this, the identical copy that's attached is called the chromatid, or specifically the sister chromatid. So in this picture, I have 4 chromosomes, 1, 2, 3, 4, 2 pink, 2 green, and each of them has an identical copy attached. So if I was going to represent this cell, I would say, oh, I hate writing on this. I would say 2n equals 4. There are 4 chromosomes. Now, how do I know this cell is 2n? Well, remember I said somatic cells do this, and unless otherwise specified, somatic cells are 2n, diploid. They have 2 copies of each chromosome always. So if I look These two chromosomes here are kind of longer, and these two are kind of shorter. So I would call the longer ones chromosome number 1. I have 2 copies of it. And I would call the shorter ones chromosome number 2. I have 2 copies of that. So that is how I have 2n equals 4. Now I could have seen this in prophase. I just like metaphase because they're lined up more beautifully here and it's easier to see. Now does anyone want to ask anything about metaphase? Yes, Rody.
Is this like the other 10%? Because you said
Yes.
phase is 90%.
This is the other 10%. So cell division is the other 10%. Mitosis will make up most of that, and then there's also that thing I showed you on the slide that's called cytokinesis. But together they make up the other 10%.
So this would metaphase is just 10%.
Not just metaphase, prophase, metaphase, and then the A and the T that we haven't got to yet, and cytokinesis together make up the other 10%. Okay? Anyone else have questions? We're getting to the end. Anaphase. Anaphase, so that's the A in PMAT. The key features in anaphase are first that the centromeres divide. So instead of the 2 copies of the chromosomes being attached by the centromeres, remember a spindle fiber was attached to each of them. And so the spindle fiber is going to pull the chromosome to the side that it came from. So if you look, the chromosomes don't look like straight lines anymore. They kind of look like V's or A's if you're looking at it from the other direction. Imagine that you were pulling something from the middle. The middle would go first and then the 2 sides would follow. That's why the shape of the chromosome has changed, just because they're being pulled. Because the centromeres divide, the sister chromatids separate. So, like I said, I had 4 chromosomes on my picture. I had 2 identical copies attached by a centromere of each of them. In anaphase, I'm pulling those chromatids apart. Now the way this works is that the spindle fibers shorten, and as they get shorter, they pull the chromatids to each pole. Now often, but not always, in pictures, this one represents it. You'll see an elongated cell, so it'll look like the cell is stretched out. This is the thing that I find gets left out of pictures the most, especially pictures that I've seen, say, on older diploma exams. Sometimes they don't represent that the cell looks stretched out in anaphase. So please don't use that as the only factor telling you it's anaphase. It can certainly be one of them. But if it's not elongated and the other things are happening, you're definitely still looking at anaphase. Now we have one more phase of cell division. Does anyone want to ask anything about anaphase before we go there? So the last phase of mitosis is telophase. In this phase, so it's the T in PMAT, the chromatids will start to unravel and form chromatin. So you might look at this picture and say, those still look like chromosomes. I can count them. And so keep in mind that these features that I'm telling you are what will happen once the phase has totally completed. So we have a tendency as teachers to draw the chromosomes in telophase still condensed like it's at the very beginning of the phase so that you can count them, so that you know how many chromosomes there are. If we waited until the very end of the phase, instead of seeing 4 distinct chromosomes, I would just see jumbly mess inside of the nuclear membrane like I did in interphase. Now the next thing that happens is the nuclear membrane reforms. Now this picture shows it already fully complete, but keep in mind this is what's happening as this phase progresses. So you might also see it as a dotted line instead of a full complete nuclear membrane. The thing that I think I would say universally shows up in pictures is this. In animal cells, it's not the same as plants, but in animal cells, there is very, very clear, distinct pinching. Sometimes you can kind of see it in anaphase, but in telophase, you can definitely see it. It looks like an 8, right? It's very pinched in the middle. It's very obvious that something is happening and that we're starting to divide. Now, at the end of this phase, if we were to count the chromosomes, there are 1, 2, 3, 4 on one side and 1, 2, 3, 4 on the other side. There are still 4 chromosomes. It's the same as the number I started with. All the cell did was make an identical copy of each chromosome and then split them apart. That's how we made the new cell. So in the end, I end up with 2 cells that each have 4 chromosomes, just like the one I started with. Now,